Okay, so in this video, we are going to review Unit 6 for AP Biology, which is our unit on DNA, RNA, gene expression, and gene regulation, or basically molecular cell biology. All right, so each of these slides, when you see words at the top, these are the enduring understandings and essential knowledges from the AP Biology framework. So our first one we start with is DNA, and in some cases, RNA is the primary source of heritable information. So when we look at DNA and RNA, we have a couple different, well, we can look here. So a couple different ways that genetic information is transmitted from one generation to the next. So when we look at DNA as our genetic information, you have the linear chromosomes that you find in eukaryotes that is passed from one generation to the next through meiosis and sexual reproduction. Then we also have prokaryotic DNA that includes includes their like circular chromosomes that are passed from one bacterium to like the next generation through binary fission. So here in the middle, this GIF that you see is basically how like a circular chromosome will go through DNA replication. And then over here on the right, this is a GIF of binary fission that we see happening. And we could also have a type of reproduction called asexual reproduction, which is basically reproducing by mitosis, which would use linear chromosomes as well because it's found in eukaryotes. When it says in some cases RNA molecules, that may be referencing like RNA viruses, I guess. Okay, so prokaryotes and eukaryotes can contain plasmids, which are small extra chromosomal double-stranded circular DNA molecules. So when it says extra chromosomal, that means like not part of the main like chromosomal DNA. So here in this prokaryote, you see the nucleoid region. That is the one like main circular chromosome that you find in prokaryotes. Now that green circle is the plasmid that is like separate from that, but it's like a tiny circle of DNA. It is a double helix and two strands, but in like a circle fashion. Now we also find these plasmid DNA in eukaryotes, in our mitochondria, and in chloroplasts. And this is because mitochondria and chloroplasts evolved from ancient prokaryotes millions of years ago as understood by the endosymbiotic theory. And so in like humans, for example, our plasmid DNA is passed in mitochondria from the mother to her offspring. It's like a maternal line of inheritance. There are genes on our plasmids and they can be expressed and they are non-nuclear DNA or like outside of the nucleus. DNA and sometimes RNA exhibits specific nucleotide base pairing that is conserved through evolution. So A pairs with T, C pairs with G. This was discovered by Chargaff in the 1940s, I believe 1947, as he looked at numerous or different DNA samples from a wide range of species. He found that the amount of A pretty much always equaled the amount of T, the amount of G equaled C. And when we categorize these nucleotides, we find that G and A have a double ring structure, and we call those purines. And then T and C and U, uracil and RNA, would have a single ring structure, and those are your pyrimidines. Now, if you ever have to memorize these for any reason, the way I remember it is that a T and the C, thymine and cysteine, both have Y's, and so does the word pyrimidine. I don't know if that'll help you. Okay, so DNA replication ensures continuity of hereditary information. It ensures that like a complete set of DNA will be passed from parents to offspring, or even from one cell to the next through the cell cycle. So when we look at DNA replication, we start out with a double helix, And basically, DNA replication happens in S phase of the cell cycle. So you can look back at Unit 4 in in AP Bio about S phase of the cell cycle and making sister chromatids. So right here, as we watch this GIF, you start with a single strand of DNA and it unwinds. We'll see an enzyme called helicase do that. Now, each strand, once it's been opened, will serve as a template to build the complementary strand according to base pairing rules. So you can see right here, you have the original strands on the outside, and the new complementary strand is being built on the inside. So if you know one side of DNA, you can figure out the other. So this is where the term semi-conservative comes from, because the original DNA strand that you see right here is semi-conserved. Here we have these two daughter strands that were made, and they're half of the original. Let's see if I can write on this. So like here you have half original, and then on this side you have the new complementary sequence being built. So the DNA is like semi-conserved, half original, half new. And when we look at how, so here is another GIF kind of showing this. So you unwind the double helix, and then you can do base pairing rules. If there's a T, then you'll pair an A, etc. So if we look at the enzymes involved in DNA replication, our first enzyme is going to be helicase. Well, technically, there'll be an enzyme called topoisomerase. So topoisomerase would kind of be like in this region, kind of being to help relax or loosen the DNA to make it easier for helicase to break those hydrogen bonds. So here you have hydrogen bonds that are responsible for base pairing A with T, C with G, and helicase will unzip that. And then in order to start replication, their DNA polymerase 3 will actually need what's called a primer. And that primer is made out of RNA nucleotides. So an enzyme called primase can come and build a primer. And you know this is RNA because it has a U in it. And then from that RNA primer, then you can have DNA polymerase 3 can begin to extend and build that new strand of DNA. So when we look at how the strands are built, though, DNA polymerase 3 builds in an anti-parallel direction. It builds from the 5' to 3' direction. And when we look at how that works, once you have that RNA primer attached and built, then DNA polymerase 3 can come and begin base pairing A with T, C with G, and it grows like DNA polymerase 3 can only add to that 3' end. So that first part, this area right here will be the 5', and as it grows, it's adding only to that 3' end. If you want a more detailed explanation to this, I highly recommend my video on DNA structure in my Unit 6 playlist. I spend a lot of time looking at the nucleotides and talking about anti-parallel and 5' and 3' directions. It'll be much slower, more detailed than just looking at GIFs. Okay, so now though, once DNA replicates and builds a complementary sequence, that is used for DNA replication in the cell cycle. So before a cell can divide, it needs to replicate the DNA, and that would be with DNA replication in S phase, making your sister chromatids that are identical copies, and then they'll be separated during mitosis or meiosis if you're making gametes. But our DNA is like our genetic information and our genetic sequences that are going to be responsible for carrying our genes. And so when we talk about our genes and how they code for our phenotypes, it really comes down to the process of expressing those genes. And expressing those genes means taking that DNA sequence and ultimately producing a protein. So that process is called protein synthesis and involves 3 types of RNA. So our first type of RNA is called messenger RNA. And its job is to carry the information from our DNA. So our DNA is our directions on how to build a protein. Well, that messenger RNA is going to leave the nucleus where it'll find a ribosome. And a ribosome is your next kind of RNA called rRNA. Ribosomes are basically made out of rRNA and proteins. And that ribosome is where tRNA, the third kind of RNA, tRNA will read the messenger RNA in groups of 3 called codons to bring the appropriate amino acids to build the protein. So our genetic information flows, this is kind of fast, GIF, sorry, from a sequence of nucleotides in DNA to a sequence of bases in messenger RNA to a sequence of amino acids in a protein. So I tried to color code my words with what you're seeing in the GIF. So you start with your DNA sequence, and then a messenger RNA is transcribed. And then from that messenger RNA, it has the directions. The ribosome you see here is green, and that will read the messenger RNA to build the protein. This is called the central dogma of biology. So how this works in protein synthesis is RNA polymerase will use just one side of the DNA. RNA is single-stranded, so we only are going to use the template strand of DNA to direct the building of messenger RNA. So like the sequence of the RNA will be based on the sequence in the gene. So here you can see RNA is also built from 5' to 3'. You can see the nucleotides are only added to that 3' end. And in this background, RNA polymerase is green, and DNA is only opened to do transcription where there's RNA polymerase. Once the RNA breaks off of the DNA will actually wind back up into a double helix. So this process right here of building a single strand of RNA is called transcription. In prokaryotes, it happens in the cytoplasm, but in eukaryotes, it occurs in the nucleus. Now, like a miswording that I see a lot year after year is that students will like write or say the DNA is turned into RNA. False. We did not turn DNA into RNA, but rather we use the DNA as a template or a guide of how to build the RNA. And that DNA is going to be safe and in the nucleus and can be used over and over again. And then the RNA is really just a copy of that DNA. Okay? And so here you can see how the DNA is opened. And then the one side, the template side, is used to build the messenger RNA. But there's also other words you may see, like the coding strand, the non-coding strand, and the antisense strand. Okay. So the enzyme responsible for synthesizing messenger RNA or making messenger RNA is called RNA polymerase. And if you remember from Unit 1, poly means many, or a polymer is a chain. So here, this is a chain of RNA nucleotides. So that's where the name, and then ACE stands for enzyme. So here you have like an RNA polymerase. And it's built in the 5' to 3' direction. And it basically is using base pairing rules to build the RNA. Now, because RNA and DNA also run anti-parallel, the DNA strand, like how do you know which strand out of the 2 will serve as the template? Well, if RNA is built from 5 to 3, that means that the DNA needs to run from 3 to 5. So that's important in figuring out, if you have it like written on paper, like which strand of DNA to use. The DNA and the RNA will run anti-parallel to each other. But now this is something like special and unique that happens in eukaryotes. Eukaryotes, their process of transcription, which this is transcription, occurs in the nucleus. Prokaryotes don't have a nucleus, so here we're only talking about eukaryotic cells. They actually are going to undergo a process called RNA processing, where we take the pre-mRNA that's made and we edit it. This is actually pretty rad because it helps to produce multiple different proteins from one gene, actually. So here we go. The first step is to identify introns versus exons. Now, you could get confused on this, but I want you to think about exons as the region that is expressed in gene expression. Like, that's the important part. And then introns are basically just in the way. They're in between the important parts, so we want to cut them out. So what you can see in this GIF here is we're going to start with a or a primary mRNA. And here we have exons that are numbered, and then the light yellow are the introns. So we're going to splice out introns and then splice together the exons. So this is how, if like a word problem or something says a gene in DNA is 12,000 bases long, but then the mature mRNA is 8,000 bases, like how is that possible? Well, those 4,000 bases were all bases, nucleotide bases, A, T, C's, and G's, that were in introns, so they were removed and spliced out. So if you ever see like a context where the gene in DNA is longer than the mature mRNA, that has to do with the splicing out of introns. If a mutation happens in an intron region, it's going to have zero effect on the phenotype because it's spliced out. Even if it's like a frame shift style mutation, because even if you delete a little bit of intron, the exons are, like, think about this. If you have like a mutation, let's say there's a deletion right here in that intron, and then the introns are spliced out. Well, who cares if there was a shift? The sequence in like number 3, for example, and 4 and 5 and 2 are still the same sequence being spliced together as the final product. Okay. Other things that happen in RNA processing is the addition of a 5' cap. It's a modified gene nucleotide that will basically help the mRNA to attach to the ribosome to start translation. There's also a polyadenine, poly-A tail, a bunch of adenine nucleotides added. That poly-A tail could be anywhere from 50 to 250 adenines added at the end of the messenger RNA. And this is basically so that once the messenger RNA is in the cytoplasm, the enzymes that are there that degrade mRNA and hydrolyze mRNA. Remember, hydrolyze means to add water and break apart. So as mRNA is degraded in the cytoplasm, that poly-A tail extends the life of the mRNA in the cytoplasm. So the longer the poly-A tail, the longer it can last and be expressed. And we see here in pink part D, where we talk about alternative RNA splicing. Basically, a gene that codes for a protein, like when they discovered, like in the last few decades, when it was like a single gene codes for a single protein hypothesis, that was disproven when we discovered that the human genome has about 25 to 30,000 genes, but in reality, there's over 100,000 proteins. So if one gene coded for only one protein, you would need over 100,000 genes, and that's not the case. And so we can edit the RNA in different ways. So here, you can change what is an exon, or like you can cut out different exons at different times, which would give you different RNA products. So if you would like more information on any of this, I have more videos that go into detail on each of these things. But I also really like this GIF because it shows the spliceosome, and how we cut out the introns. So a spliceosome is made of multiple SNRPs. One of these purple things is called a small nuclear, like ribonucleic protein, I think, a SNRP. And so it basically, multiple SNRPs will join to this location between an intron and an exon. And when they come together, they form what's called a spliceosome. And they splice out that intron, which is orange here, and the two exons are spliced together. Okay. So translation now of the mRNA to generate a polypeptide occurs on ribosomes that are present in the cytoplasm of both prokaryotic and eukaryotic cells. So all cells have ribosomes. However, it's only eukaryotic cells, sorry, I said that weird, eukaryotic cells that also have ribosomes on the ref-ER. But regardless if a ribosome is on the ref-ER or found in the cytoplasm, they both read the messenger RNA, which we see here, codon by codon, where the transfer RNA, the tRNAs, will bring the appropriate amino acid, building the polypeptide. So ribosomes are like your little protein factories, regardless of where they are located. So depending on your teacher, some teachers may go into more detail on this, but you can see right here on this GIF, this right here is the A site. If your teacher references different sites on the ribosome, the A site is where the arriving tRNA carrying the next amino acid is in the ribosome. Then you have the P site, and that is the part of the ribosome where it's holding the growing polypeptide. So you can see right here, like a peptide bond forms between those polypeptide, or between the amino acids. And then the tRNA moves from the A site into the P site. And then eventually it's in the E site, which stands for exit. And then it will exit. And so the next time you eat protein, let's say, like my kid just made a peanut butter and jelly sandwich, the protein in that peanut butter, his body will digest and have free-floating amino acids that then can go and attach to new, not new, but empty tRNAs and replace the amino acids that were used in building this polypeptide. Now, when we compare prokaryotic cells, I speak weird, prokaryotic organisms to eukaryotic cells and how they do protein synthesis, there's some differences. The first difference is the location. For eukaryotes, we have transcription happens first in the nucleus, and then translation, which is building the protein, happens second in the cytoplasm on a ribosome. Whereas in a prokaryote, transcription and translation happen simultaneously. Now, because it's happening simultaneously, that doesn't really give any time for RNA processing. So we, eukaryotes, have that extra step of RNA processing. We're going to cut out introns, splice together exons, add the 5' cap, and the poly A tail. So if we want to use eukaryotic DNA in genetic engineering and have it be expressed in a prokaryote, we would need to have already spliced DNA. Or mRNA, like basically DNA that doesn't have introns, because prokaryotes don't have any like RNA processing machinery or enzymes to do that. Okay, so translation has a couple of steps. You have the initiation step where you have the first tRNA will attach the start codon. So AUG is always your start codons here. Now, this region above or before the start codon is called the untranslated region, the UTR. So some RNA may have an untranslated region that is not coding for the protein, but the first tRNA carrying methionine will attach to that AUG, and then we'll have, that's part of initiation, then you'll have the small ribosomal subunit will attach, and then a large ribosomal subunit will attach. Then the elongation phase is basically what we saw in this GIF here, where we're adding and growing adding amino acids to grow the polypeptide. That's the elongation phase. And then basically the termination phase is where when the ribosome is reading that messenger RNA, it'll reach a stop codon. And a stop codon signals the end of the gene. Basically like a releasing factor will come and everything will break apart and you'll have your completed polypeptide. And that is translation. So if you wanted to pause the video and read this step by step over here on the left, well, we could do it together, I guess. So translation is initiated when RNA, which is part of the ribosome, interacts with the messenger RNA at the start codon. Then the sequence of nucleotides on the messenger RNA is read in groups of 3 called codons. This is where you'll use that chart, the genetic code, to figure out which amino acid is coded for. Now be very careful. I still have my own students make this mistake. When you use that genetic code and you look up AUG, oh, that's methionine, or UUU is phenylalanine. You want to make sure you're using the codon in the messenger RNA and not the anticodon. So let me explain. Let me change my color pen real fast. So when we look at, let's use purple here. So for example, this AAA is the codon that codes for lysine. Now, if for some reason you use the anticodon, UUU, on that table, that'll code for phenylalanine and your answer will be wrong. So you want to make sure that you're using the codon that's in the messenger RNA. The anticodon that you find here is really just to help that tRNA attach to the codon and stabilize it in place long enough for a peptide bond to form between those amino acids. Now, the cool thing is that the third base isn't super strict when it comes to base pairing rules, and that's called the wobble effect. And so really in real life, we have about 45 different tRNAs, even though there's 64 codons, because that third base isn't as strict when it comes to base pairing rules. If you want, you could look more into the wobble effect on Google if you need to. Okay, so each codon specifies a specific amino acid, but some amino acids could have more than one codon. So for example, like serine has like 6 codons that code for serine. Like that, I don't know. But like UUU will only ever be phenylalanine. Okay. The genetic code is used by nearly all organisms on Earth. It is evidence for common ancestry as part of our Unit 7 on evolution. And the tRNA brings the correct amino acid based on the mRNA codon, and it's added to the growing polypeptide chain. So this right here is actually the first step in building a protein. So if you look back at Unit 1, where we talk about carbohydrates, lipids, proteins, and how we build proteins, this is the first step. This is the primary structure of a protein. This is also why mutations can have an impact on phenotype, because when you have a mutation in the DNA, now the messenger RNA is different, and that could possibly code for a different amino acid, which changes the polypeptide and possibly how it folds up, changing the protein structure, which may change how it works, which may change the phenotype of the organism. Okay, it's beautiful how everything is all connected. Okay, and so this process continues until you get to a stop codon. So genetic information in retroviruses is a special case and has an alternate flow of information from RNA to DNA, made possible by reverse transcriptase, an enzyme that copies viral RNA, a genome into DNA, and this DNA integrates into the host genome and becomes transcribed and translated for the assembly of new viral progeny. So I'm going to back up and we're going to start that again. So here you see a virus, and this is a retrovirus, such as maybe HIV or something. So you can see how it enters the cell. Now, the red here is the viral mRNA, and the black dot is the enzyme reverse transcriptase. So this reverse transcriptase will do transcription in reverse, building a single strand of DNA and then using that one single strand to build the complementary sequence. So now you have a double helix, and now that double helix will incorporate into the host's genome, where now it will be transcribed and make more copies of that viral RNA, and then spread the virus. So yeah, man, I have a video on how viruses work, but retroviruses are a trip. And because that there's no proofreading or proof checking that this reverse transcriptase, like to see if it made any mistakes here, this is one of the reasons why like retroviruses mutate, have so many mutations from one generation to the next. There's no like checking for errors. Okay. Ooh, regulatory sequences are stretches of DNA that interact with regulatory proteins to control transcription. Okay, so we have regulatory sequences in both prokaryotes and eukaryotes. So when we talk about eukaryotes, we have that enhancer region that helps turn on gene expression. It has a role in gene expression in eukaryotes. And then in prokaryotes, in bacteria, with their operons, their regulatory sequence, they have the step number 1 that codes for repressor proteins that can attach to operators or not. So we'll talk a bit about these, but I have a whole video on eukaryotic gene regulation as well as a video on prokaryotic gene regulation with operons and stuff. I do recommend that you watch those and have a good solid foundation on gene expression and regulating gene expression. Okay, so let's go ahead. So the top here is eukaryotes and the bottom is prokaryotes. We also have epigenetic changes can affect gene expression through reversible modifications of DNA or histones. So with epigenetics, we have histone acetylation that can cause the chromatin to unwind or loosen, which would be called euchromatin. And then we have methyl groups can also be added to our DNA, which would create heterochromatin. So when DNA is methylated, it basically is turning off gene expression. You can watch this more in my eukaryotic gene regulation video. And then we have the phenotype of a cell or organism is determined by the combination of genes that are expressed and the levels at which they are expressed. Okay, so your phenotype is based on proteins, the presence or absence or type of protein you produce. Now, how do we produce those proteins is based on expressing those genes. And this starts when we are like a fertilized egg, a zygote. When I guess like here we can go with stem cells. So as soon as a sperm swims into an egg and you're one cell big, like that has your like entire genome. You got your 23 chromosomes from your mom, your 23 chromosomes from your dad. That now every single one of those cells has your 46 chromosomes. And I think if you count, this is actually 23 pairs. The purple ones represent like maternal, the green ones represent paternal. So like in your stem cells, it has your entire set of DNA. And as your cells divide, they differentiate or they become different. Every single cell in your body has the exact same DNA, except for your red blood cells. They actually will get rid of your DNA. They don't have a nucleus. They like get rid of it after it's made. But basically, every single cell in your body has the exact same 46 chromosomes, but they are different because they produce different proteins. So if we go back to like here, observable cell differentiation, it's because in the early stages, so like here we have like the lens of your eye being formed. So as your cells differentiate from a little embryo, the cells that are going to form your lens will express the protein like crystalline or something, I think it's called. I don't know how to pronounce it. I know how to read it. That is only found in your eyeball cells, like in your lens cells. However, the cells in my elbow also have the DNA to make my eyeball lens, but they're not expressed in my elbow. They're only expressed in the area that will develop into a lens of an eyeball. So when we talk about expression of genes for tissue-specific proteins, those like transcription factors, so like in this developing eye, there'll be transcription factors to turn on those genes to have the lens develop. The transcription factors for eyeball development, you don't find in my elbow. You have the DNA and the genes, but not the transcription factors. It's transcription factors that are super important in regulating our gene expression. So when we talk about like morphogens or cytoplasmic determinants, basically as our tissues are developing, we have different transcription factors we are exposed to that will turn on tissue specific proteins. It'll turn on gene expression. So like here on the left, up here at the top, you have a high amount of like morphogens or transcription factors to turn on gene expression, whereas over here, you're missing those transcription factors and the genes will not be expressed. So you have different gene expression depending on which transcription factors are present, and that will lead to different cell types. So here in eukaryotes, when we talk about transcription factors, we have specific ones that are going to attach to our enhancer regions. And when they attach to our enhancer regions, these specific activators, well, then that's going to join with some general transcription factors to attach to our promoter region that allows RNA polymerase to now attach and begin transcription. This video is already pretty long, and I do feel like if you're weak in this area, please watch either mine or other videos on this. It is super important to understanding the big picture. Now, prokaryotes, they have groups of genes called operons, and they also have a promoter. However, they don't use specific transcription factors like we do. Instead, they use repressor proteins like we saw, oh goodness, kind of a far way back. They use like repressor proteins that will then be involved in turning on or off gene expression in their operons. And so prokaryotes have inducible operons, which would be like your lac operon, or they have repressible operons like the trip operon. Okay. Promoters are found in both prokaryotes and eukaryotes, and they are upstream or before the gene, and that is where RNA polymerase begins transcription. It is also where transcription factors bind in eukaryotes to help begin transcription. Okay. And in bacteria or prokaryotes, you would find they do have a promoter, but in prokaryotes, they would have what's called an operator here that would also help with regulating that transcription. So in eukaryotes, which is us, we have this enhancer region that is upstream. So if here's my promoter, which is where my RNA polymerase will attach and begin, upstream or before it, you have the enhancer region where we have our very specific transcription factors that will activate or repress and attach that enhancer sequence, leading to the DNA bending over and bringing everything together into a complex to begin transcription. But there can be negative regulatory sequences or mechanisms that will inhibit gene expression. So in prokaryotes and bacteria, they could have a repressor protein that will block RNA polymerase, and that operon is off. In us, we could have repressor transcription factors that will block that enhancer region and prevent transcription of a target gene. So we also have ways to turn on gene expression, but also inhibit gene expression. Now, gene regulation and controlling which genes are expressed results in differential gene expression. And that will influence which proteins are made in a cell. So it's super important to remember that in like a eukaryotic organism, we are trillions of cells. Every single cell in your body has the exact same DNA, but they're not all, or not all of your genes are equally expressed. It comes down to your transcription factors, your DNA methylation, your histone acetylation, etc. And turning on and off genes will lead to different proteins in a cell and lead to that cell specializing. Okay. Now, when we talk about gene expression, one way you can measure gene expression is through protein products. And so if transcription already occurs and messenger RNA is in the cytoplasm, one way we can block translation is with the use of microRNAs, small RNA molecules that can actually, like in green here, can actually base pair with an RNA molecule, and it will block translation, preventing a protein from being made. So in the old days, we used to call a lot of our DNA junk DNA because we didn't know what it coded for. Turns out some of it's coding for microRNAs. Now moving into mutations. So alterations or mutations or changes in DNA sequences can lead to changes in the type or amount of proteins produced and the consequent phenotype. So right here, I want to connect this over to Unit 4. So the type or amount of the protein produced and change the phenotype. So sometimes you could have a mutation where you are overproducing a protein, and that's one way that can lead to cancer. If you're overproducing like growth factors or the receptors that receive growth factors, or even like the relay proteins or the cyclins that control the cell cycle, sometimes like mutations in those important genes can lead to confusion in the cell cycle, which lead to uncontrolled cell growth. Okay, but also other alterations in DNA, for example, changing like the phenotype. If you have a mutation that prevents like melanin from being formed. Melanin is our like protein that gives us pigment in our skin. So if you don't make melanin, then your phenotype, you'll be albino. Like you won't make melanin, and therefore you won't, that'll affect your phenotype. So anyway, when we look at mutations, though, we have different kinds. We can talk about point mutations. We have 3 different kinds of point mutations. The first one is called silent, where you can have a mutation happen. Let's look at the second codon here. So the original one was an A, but a mutation occurred where now it's a T. That mistake or mutation will happen during DNA replication. So in S phase, when you have your DNA strand and the double helix is opening, you're using each side to build the complementary sequence, to build those sister chromatids, there can be mistakes, and DNA polymerase can accidentally attach the wrong nucleotide. This happens about 30 times for every cell division. However, only 1% of your DNA is protein coding or a gene. So 99% of your billions of nucleotides don't code for your proteins or your phenotype. So most of your mutations are going to be neutral, like they're happening in non-protein coding areas. Now, it could happen in a gene, but a mutation could happen in an intron and then have no impact. Okay, so a silent mutation, though, if we look at that second codon, it's the first amino acid is valine, and it's still valine here. Now, a missense point mutation is where in this third codon, instead of a C, you see it is an A instead, and that changes the amino acid from proline to threonine. And that would could have an impact on how that protein folds. So if this mutation happens, especially like in an active site of an enzyme, that could have an impact on whether or not the substrate attaches. So kind of depending on where mutations occur in that polypeptide can also have an impact on how it folds up. You could also have a nonsense mutation, And in a nonsense point mutation, it's a stop codon that's created. So a stop codon, basically, if that happens like mid-RNA or early RNA, when the ribosome gets to that stop codon, it stops. And at that point, it is the polypeptide is finished. So it like no functional protein is made. Now, I want to point out, when you look at these mutations here, we're looking at the DNA, and you might be thinking to yourself, why are there T's that code for like valine, for example? Well, in actuality, this would be like G pairs with C, T pairs with A pairs with U. So the codon would be CAU in the mRNA that codes for that valine. But the mutation happened in the DNA that then changed what the mRNA is that could change the amino acid. So if you ever asked like what mutation caused this change, you would figure out, you'd work backwards, start with the RNA and go back to the DNA, and what was that mutation in the DNA. Okay. Now, sometimes we could have things that are called frame shift mutations. Okay, sorry, I had to pause there for a second. I think I was talking about frame shift mutations. So in a frame shift mutation, this is where you're going to have an insertion or a deletion. So sometimes like extra nucleotides can be added on accident or removed, and that would shift the entire sequence one way or another. That's why it's called a frame shift mutation, and that would change the codons. And therefore, changing the codons will change the amino acids, which changes the protein. So I encourage you to like write out a DNA sequence and then transcribe it and translate it, and then erase one or two of the DNA and do it again, and you'll see how you get a totally different polypeptide. Now, a type of insertion or deletion that wouldn't be as bad is if it is like in a group of 3. So if you insert 3 nucleotides together, well, then that would be just like inserting 1 amino acid, and it may not have a disastrous impact. Or if you delete 3, you're deleting just 1 nucleotide. So after that insertion or deletion, then the rest of the reading frame would still be the same. So that may not be as harmful of a frame shift. So like here you can see like deleting or inserting would shift or change the whole reading frame. So when we look at our next standard, sorry, I'm looking for my mouse. So this one says changes in genotype can result in changes in phenotype. Now, personally, I think this is one of the most beautiful concepts in our class in biology. I love thinking about how like our chromosomes are like a record of our ancestry, and then those chromosomes like have crossing over, they mix our genes, they get passed from generation to generation, but then that sequence of DNA that we inherit has like this strand or this sequence, and that DNA codes for our proteins, and then those proteins are what give us our phenotype. So when we talk about this phenotype, then our phenotypes are acted on by natural selection and survival of the fittest. So here, the function and amount of gene products determine the phenotype of organisms. And when we look at a famous, popular example, I don't really know what the right word is, it's sickle cell anemia. So when we talk about our protein called hemoglobin, that's responsible for transporting oxygen in our red blood cells, hemoglobin is a protein, and it has this kind of like round shape that you see up here. And some individuals, there's a mutation that some people can inherit right here, where 1 nucleotide, so this is a point mutation, and it changes the original amino acid from, I think it's glutamic acid, to valine. So this would be a missense mutation. So the polypeptide is still made and produced. However, those R groups that influence the folding up in that secondary and tertiary structures and the quaternary structure, are now different. And now, instead of like a round hemoglobin shape, this mutation causes the protein to be more straight, all from one change in one nucleotide, one mutation. And this causes a sickle cell shape, which makes the red blood cells like less flexible, harder to carry oxygen, more difficult to flow in the blood. So when we talk about this, a mutation can either be like detrimental, beneficial, or neutral, but it really depends on the environment and the context of that environment. So while inheriting sickle cell anemia is detrimental to your survival, like a low fitness, having 2 copies. So let's look over here at this blue family. This girl here, it says affected daughter. If she inherits 2 copies of that sickle cell allele, then all of her red blood cells are going to be sickle shaped. And while she's resistant to malaria, this is fatal for sickle cell because her red blood cells just really can't function as the body needs. Now, a person, so if we talk about the environmental context, in regions of the world where you have malaria, malaria is a parasite that enters into your red blood cells and multiplies within red blood cells. And about every 48 hours or so, the red blood cells burst, and that malaria parasite gets out and infects other cells and repeats its life cycle over and over again. So when we look at this environmental context, a person who's homozygous dominant, like no sickle cell mutation, no sickle cell allele, and all of their red blood cells are round, while they'll be healthy at transporting oxygen in an area where there's malaria, they are susceptible to dying to malaria. Versus a person who is heterozygote for this trait, like how it says like a carrier. So half of their chromosomes that are expressed, or half of their red, like hemoglobin genes, are carrying like the mutation for sickle cell, and then on the other chromosome is the mutation, or no mutation, like normal hemoglobin. So some of the red blood cells will produce like a sickle shape, and some will produce a round shape. So this person actually has what's called like a heterozygote advantage. And in areas where there's malaria, they survive malaria because they don't have enough round red blood cells for this parasite to be successful. But so they're resistant to malaria, but they can also transport oxygen. They have enough round red blood cells for that. So here, this phenotype is selected for in areas where there's malaria, but here in like Southern California, we don't have malaria as a like environmental pressure, so therefore this would not be selected for, right? Like then this phenotype would be high fitness. So it depends on the environment. And mutations are the primary source of genetic variation, so that is true for both prokaryotes and eukaryotes. Mutations create variation. Like the first person to have blue eyes was due to a mutation 6,000 years ago. And if blue eyes didn't harm you, your reproductive success, or help you, then that would be a neutral mutation that spread. So scientists currently believe that, I hate the word believe, the current hypothesis is that every person alive today with blue eyes has that same genetic mutation. Like blue eyes didn't evolve multiple times. Now errors, I do want to talk about this standard a bit because I noticed some of my students made mistakes when I had it in class on like a FRQ or something. So errors in mitosis or meiosis can result in changes in phenotype. So you see here where it says changes in chromosome number can result in new phenotypes, including sterility caused by triploidy or increased vigor of other polyploids. So polyploidy you often find in plants, which we talk about this when we get to Unit 7 and we talk about post-zygotic, pre- and post-zygotic barriers for what is a species. But basically, sometimes like plants can have mistakes in meiosis where their gametes have a whole extra set of chromosomes, or maybe like 2 sperm fertilize the same egg, or something where they have a completely extra set of every kind of chromosome. Like that would be triploidy, and that should result in being sterile. But polyploidy can result in like a tetraploid organism or something where they have 4 of every kind. And that could create a new species if it was able to self-fertilize like in plants. Now, that is different than the results of non-disjunction. So in non-disjunction, which was the last slide of Unit 5, so if you're unfamiliar with non-disjunction, just hop to the very end of Unit 5's video. But non-disjunction is when there's errors in meiosis, and it results in just 1 extra chromosome, like in Down syndrome. So in Down syndrome, it's just 1 extra 21, not triploidy. They have 2 of every other kind, except for number 21, they have 3. Or sometimes you can have extra sex chromosomes, like that would cause like Jacob syndrome or Klinefelter syndrome, Turner syndrome, and that also is due to non-disjunction with extra X's or Y's. And so that is not the same as triploidy. That's just 1 missing or 1 extra chromosome, not an entire extra set. Okay. Changes in genotype. We're almost done. Oh my gosh, long video here. Changes in genotype may affect phenotype that are subject to natural selection. So as I talked here, this heterozygote would be favored by natural selection depending on the environment if there's high amounts of malaria. And then genetic changes that, I guess you could also say, like, if you're talking about, like I talked about albino earlier, that would actually be low fitness. Not having pigment puts you at high risk for skin cancer. But if you're an animal, you also aren't camouflaged. If you're a predator, it's hard to sneak up on your prey. If you're an albino prey, it's hard to hide from your predators. So that could also be subject to natural selection. But here we talk a bit about genetic changes that enhance survival and reproduction can be selected for by environmental conditions. So this really is like Unit 7 and natural selection. But here we talk about Part A is talking about prokaryotes and how they can acquire genetic variation that can then be acted on by natural selection. So bacterial transformation is when bacteria have the ability to take up naked DNA from their environment. So for our example, if they have the off chance of taking up like a gene that codes for antibiotic resistance, and now that is brought into them, that would give them high fitness and a good chance of surviving and reproducing in the presence of antibiotics. So that is an example. You could have transduction occur where a phage, a bacteriophage virus, accidentally like transfers bacterial DNA from one bacteria to another. But you could also have conjugation where a bacteria can create what's called a sex pilus, or like a tube or a channel that connects the 2 cytoplasms together. And then that plasmid can be transferred from the donor cell to the recipient cell. And then there's transposons, which are like jumping genes, where genes can move locations in a chromosome or in a DNA sequence. So if we go back to this Part A, that's talking about different variation and ways that bacteria or prokaryotes can acquire variation separate from just mutations. And then that new variation can be acted on by natural selection. Now for Part B, related viruses can combine or recombine genetic information if they infect the same host cell. Viruses don't really follow the same rules that cells do because viruses aren't alive. So you can have, like, if you have 2 viruses infect a cell, their DNA can recombine and make a new genetic information, And then Part C, reproduction processes that increase genetic variation are evolutionarily conserved and are shared by various organisms. That's referencing like your crossing over, independent assortment, random fertilization, basically sexual reproduction, because it favors increasing variation in populations that undergo sexual reproduction. That would be selected for and then spread and be shared evolutionarily. Our last slide in this, or our last topic, is genetic engineering or biotech. The first one, part A here, talks about gel electrophoresis. So let's just see that for a minute. So in gel electrophoresis, this will be used to compare DNA sequences. So if you take a strand of DNA or a sample of DNA, you can cut it with a restriction enzyme, and then it cuts it into different size fragments that you load into a gel. And because DNA has a negative charge, you actually will hook this up to like a machine that has a power supply with a negative electrode on one side and then a positive electrode over here. And with that, when you turn on the power supply, your DNA is also negatively charged. So that DNA is going to move, like negative repels negative. It'll move away from the electrode, and it separates out based on size. So the smaller fragments will travel the farthest, and the largest kind of get stuck in that gel. So when you see like a banding pattern like this would be your smallest fragment, and then these would be your largest fragments. I also want to point out that gel electrophoresis can be used to compare samples. Maybe you're looking at a crime scene or a paternity test, or like comparing like fossil DNA to like living species. So can be used for comparison. And then we have, let's look at our other standards here. Polymerase chain reaction is where basically you start with a piece of DNA and then you're able to amplify it like a chain reaction. You can start with one, like a piece of DNA maybe from a crime scene or something, or maybe just a gene that a scientist is working with and needs lots of copies of, and can amplify it through like a heating and denaturing technique. And you can get from one DNA to like 1 billion in about 4 hours. So it's able to like mass produce sequences of DNA to use in genetic engineering. And then we have part C, plasmid-based transformation, where scientists can use genetically engineered plasmids. Let's pretend it's for like, let's say you're a scientist and you want a bacteria to produce human insulin because it's cheap to grow bacteria and they can mass produce insulin that we can give to diabetics. So you can genetically engineer a plasmid to have a human gene that codes for the protein insulin, incorporated into a plasmid, and then hope that your bacteria will take up that plasmid. That's called plasmid-based transformation. And then that bacteria that has that plasmid with your gene can then multiply and multiply and multiply. Then you have a whole like agar, petri dish, growing colonies of bacteria that are producing insulin for us, or human growth factor, or any other human protein that we could use in medicine or in technology, etc. So check out the video on plasmid-based transformation if you need more background on that. And then part D, DNA sequencing determines the order of nucleotides in the DNA molecule. And that is it for Unit 6. Great job. Good luck on your AP test or your unit test. And I really wish you the best and good job. Oh, good job sticking it out. An hour-long video. Dang, yo. Great job.